Okay, it's 3 o'clock, so it's time to start our next installment of Polish NMR series seminar. So this time we have a speaker from far Texas, Łukasz Jaremko from University of Texas Medical Branch, and he will give us a talk, The Age of Structural Plenty, NMR-guided chemical control of functional conformational states. But before that, I would like to remind our students that we have still best student question competition, and please ask intelligent question to our speaker. You can get a very nice set of Polish Chemical Society gadgets. As you can see here, the previous winner already got one. So be prepared and ask good questions. And with that, I'm now giving the stage to Łukasz. So thank you for joining us. And I'm stopping sharing. And the stage is yours.
Okay, let me start sharing. So, the most common and boring question since 2020. Do you hear me?
Yes, we can hear you.
Do you
And we see
see
the
my
presentation.
slides? Exactly. Okay, so first, I would like to thank for this opportunity, and I'm really pleased to see that someone is taking the burden and organizes this kind of society meetings. I would like to apologize. I wouldn't be able to do it at your regular time because now it's 8 a.m. I have first coffee in hand. It's doable, so thank you for taking into account my request to move the time. Also, I heard there are students, and I'm very happy there are some gadgets from Polish Academy of Sciences, and I've seen the cat on the picture. So cat is also given for the good.
No, it was the winner's cut.
Okay, that's so cute. Okay,
This
so
is the picture I got from him.
so congratulations and let's start. So today I just wanted to take a chance because it's not always usually we go to conferences, talks, invited seminars and we have to talk really research or we want to present ourselves from the best side and the lab from the best side. Because as I already had the pleasure to meet with some people I know from the past, and I've seen some other joined. So hello to everyone, and nice to see you at this virtually. So I decided to be a little bit more casual and take this chance to describe how basically from the perspective of my scientific journey, the view on the things evolved. So the title is Structural Plenty. And of course, there are many things and one to rule them all. And I hope that after this presentation, we will believe that the most important ring is NMR, right? So of course, we are dominated today by the internet and virtual reality more and more. So young generation is like also me, I admit, I sit all the time on the screen. If it's not the phone, if it's not my screen at work, if it's not spectrometer screen or screen of ITC or anything else, or just screen in the bar like I wanted to mention with news, then it's all internet. So let's ask, what are proteins? So more or less we see that if we think from the lecture, the science point of view, proteins, okay, so I would say there are some correct answers, right? And if we look, there are some answers that are even very correct, or they are a little bit lateral thinking. So you see, it's not always too bad. So why I study proteins? So this is my system that I like the most because it's something like, protein hardware at the same time. What I mean here, it's hardware because they do the job in the body. Software because they mediate signaling, they execute, they decide what happens, what's possible, not possible. And also they get sick, they get out of function, they get pathological, so they need repair, like our computers or software once in a while. And there are also some malicious proteins, pathogenic that enter your body. So for me, it's like nice analogy to computer. And there is also some memory issue, some execution. So it's kind of nice system. Okay, can we see proteins? Well, if we just ask the Google what we saw, yes, some of the proteins we can truly see with our eyes, but it's very narrow part of electromagnetic spectrum that allows us in indirect way to see maybe even more, but what we can see directly, so the classical, yeah, it's also a protein, but not exactly the one I want to talk about today. Okay, why we need to see proteins? To be honest, uh, I don't need to see proteins. I haven't seen a protein, but I already experienced many times. I don't know if any of you has seen a protein in this kind of nanoscale, but honestly, I don't need to see a protein. I'm interested in function. So from the medical point of view, I want to be healthy. From food point of view, I want to eat good protein. From practical point of view, I want the things to work. So the function is something that is usually like the thing that tells us that something happens with our proteins. So if we speak about biomedical research, chemical biology, medicinal chemistry, and drug design, then of course we need to understand the function. And if something goes wrong, we need to modulate it. So we need to target underlying macromolecule. Very often it's a protein. Some people try also, there are some examples with RNA, DNA, but here I'm restricted mainly to proteins. So if we would be able to see the proteins, we would not need all of this, what I'm going to talk about. So between function and macromolecule and targeting, we have plenty of things happening: structure, interactions, dynamics. And of course, to see all of this, we need to have good methods that allow us to understand and describe all of this. And here the story starts. So today, basically, we live in the times of, we all know what's inflation, right? We go to the gas station, and also here in Texas, last month, from 220 to $40 per gallon, we are now around 384. So this is inflation. Of course, the same happens in science. We see that we do our studies, defend masters, we are very proud. We defend PhD, we are even more proud. And then we do 4 or 5 years of postdoc, we do our first position, we get our promotion, we do our another position. And then we realize that there is inflation at what we have done because we look back at our papers. And the same is with structures. Today, it's not difficult to get a structure. They basically come from the computer. What I wanted to say with this slide is, first thing, these are 4 major techniques today that drive high resolution protein structures. Chronologically, from left to right, how they reached atomic, almost atomic, near atomic resolution, X-ray crystallography, nuclear magnetic resonance. Here I just want to say that high resolution NMR doesn't mean high resolution structure, just because we can see below 1 Hertz splittings on the spectra. And then we have electron microscopy. And of course, thanks to these 3 methods, we by just combining heuristically all the knowledge, we can get very decent modeling results. And beautiful things today is that single lab like mine can use all of them, even if you are not deep expert in studying. So first question, why we have 4 methods essentially doing the same, giving you structures? And why we still keep them, we don't drop any of them? I mean, usually when it's like with water suppression with NMR, why we have 8 techniques, because none of them is perfect. Everyone has some caveats, some positive and negative pluses. As I learned here in US, it's never negative. It's positive plus or positive minus. So what we are doing today with all these techniques, and especially the predicting models, is we have some kind of a problem. We have plenty of structures, we collect them, describe them to incredible detail. And it reminds me a little bit like trying to reconstruct non-existing ecosystems from past times. The here on the right is from Cambrian fauna reconstruction. So we have all those fossils. Later we put them something into the context, and like the same we do with our proteins, we put them in the context. And do we know from this how those ecosystems work? We cannot see them with our eyes. We need to reconstruct them because first, Cambrian doesn't exist. It was a long time ago. And we cannot really see our proteins. They are definitely not colorful like on the picture. So what's the problem? What do we need? The answer is simple. Moving from fossils like here, that usually if you get something fossilized, so let's say you get something crystallized, you get something stabilized in solution that you see signal like in NMR, or you just freeze proteins or complexes on the grid, this is something that persists. It persists because something went wrong, nothing has eaten or decomposed it. So I understand structural biology in a little bit similar way like fossils. If you see fossil, it means something went wrong. So that might not be fully representative or functional structure. It might be something that persists. Why it's like that? Um, we are a little bit trapped in the simplified Anfinsen's rule concept. It took me also some time to understand that it's not only primary sequence. An original paper doesn't say only about primary sequence. Original paper also gently mentions the context. So we know that basically from one sequence we can end up with many states. And even these states have some degeneration of other states that are of similar energies. So what is happening with all our prediction tools and majority of structural biology tools, we are just getting something that survived on the folding pathway or just among the energetical landscape. It's good enough, but we also need to keep it in mind that there might not be the full explanation related to the function or application. So for example, drug design. So what is underlying, what is below all of this? It's in general dynamics, because as we all know, biological motions, time regimes, it's something over 14 orders of magnitude. They can be very fast. They can be like side chain rotations, enzymatic motions to bacterial motions, and even full organism motions. So it's kind of a big concept. So how we study motions. So if we put resolution we can get on the y-axis and we put time scale. No, it's meters. Okay, resolution on y-axis, and it's early. And then we put time that we study on the x-axis, and we plot our methods. Then it looks like we are even in better situation than with determining structures if we want to study. But if we just only want to have experimental techniques, the ones that are close to native conditions, only the ones that are not requiring chemical modifications or labels like paramagnetic, fluorescent labels, then we are left with essentially three methods. If we want something further of direct method that directly can measure time as a result in indirect parameters, but still directly measures, and we want something at atomic resolution, there are two conclusions, good and bad one. Good, there is still something left. Bad, it's NMR. So now I have a question. Do you know any other technique? Because that's kind of a pretty slide if you look at that. Do you know any other technique that can essentially that measure something, one thing, over 14 orders of magnitude? Ruler, if it's like meters, millimeter, three orders, something like 4, 5 orders of magnitude, I don't have idea how you can measure it. So underappreciated, right? Just and if we look at science, what holistically as a humankind we are studying, and the time scales, then we are still not the winner, NMR. Geology gets much more years, oceanography gets much more space, ecology, biology is still more powerful, molecular biology, medicine is, yeah, just to finding a context. So now we agree that biological motions are important because what we see every day, we just, to live, we need to move. Proteins also move. And this is what we are going to look with the power of NMR. So what I'm studying essentially, depending to whom I'm talking today, it's kind of kind of interesting how the science works. So if I speak with chemists, I need to usually show them this slide. I work on reaction. Oh, great, methylation. They get not really excited. And what else you study? Yeah, molecular recognition. Oh, great. And what? Yeah, like transcription factors binding to DNA and other proteins. But at least it sounds chemical. If I tell them that I study DNA damage and repair or just transcriptional control and regulation, then it doesn't work, but it works to biologists. And we speak about the same. And everywhere, structural biology, biophysics plays a role. So why it's so important? For many years, we were and we are under the charm of human genome projects, genetic sequencing, but it turns out that at the end, all this information is essential. But how many times per lifetime you change your genome? And usually, if you do it, it's nothing good behind, right? So what changes faster, what moves faster, it's likely more and more related to life processes and things that happen. So what is important about chromatin and genetics is how often you expose and you also regulate chromatin that you allow diffunding, moving around, being recruited transcriptional factors to just express the genes, clusters, and promote changes and development in the organism. So in general, that's a simplified still scheme of organization of chromatin, and we see that there is plenty of space for really multi-level complex regulation. So now, when I've entered the topic, I usually didn't really understand much about that, what I said before, but I wanted to have my pet proteins. Of course, I wanted to have a protein that pours in the magnet, gives nice spectra, I can express purify, and my students can express purify that, and there are some effects. So what I started to do, and that I don't want to say it was visionary, it was just because proteins are like that. I started with NSD2, and then intuition told me that should be NSD1 and maybe even NSD3. But then it turned out that there is also ASH1L that should be called NSD4 because ASH2L is something totally different. But welcome to biology. So all those four proteins essentially do the same. They put two methyl groups, so methylation reaction, on histone 3 tail of lysine 36. We can ask plenty of questions about biology, enzymatic redundancy, tissue specificity, source of hyperactivity, mutations, relevance in the cancer somatic database. Okay, at the end, what's important, they are biologically relevant. And what was important for me that time is that they share essentially identical structures. So NSD1 to 3 set catalytic domain sequence identity is 80%. So they are essentially alike. There are really few residues that are different and not very much different. And what's also exciting, there are hyperactive mutations, not exciting for people who got them, but to study, that elevate the activity. So they essentially change reprogram chromatin, make it more accessible, and they make it more accessible in different tissues, a little bit different, but always with proliferation related genes that on the longer run lead to cancers. So what I want to say is if the proteins are similar, if they are almost identical, they all proteins here I show, they have the same set domain, catalytic domain, 1, 2, 3 and ASH1L are very similar, you can still get selective ligands, right? I will tell in a second how we did it, but that was actually the moment when I lost money because even I was studying NMR when I saw that from 14, 13 residues having contact to cofactor, and cofactor we explored for designing ligand, the cofactor site, that 13 from 14 residues are identical. They overlap ideally on X-ray structures between NSD1 to 3. Only one is different, and it's lysine to arginine, and that one only interacts with carbonyl backbone, so it's irrelevant. Then I said that I doubt we can get anything selective. 2 years later, okay, but it shows you something that even though that you might have small or almost no differences there are interesting things. So let's have a look at the structure of protein with our one of ligands. We can clearly see it's competitive, cofactor mimicking ligand. Adenine part is the same. We have some expansion on the left next to the chiral center. Chiral center is preserved with a amine group. And then there is something interesting. On the place of catalytic residue, tyrosine 1179 in this case, we have expansion with aromatic ring towards the autoinhibitory loop of our substituent. And then we see that, okay, that's probably the place where we can explore selectivity. Of course, we didn't know it before. We understood it once by accident. We started to have something selective. So you can clearly see that this autoinhibitory loop is kind of flexible and state dependent. If there is substrate H3 tail, it's open. When there is ligand, it's partially mainly closed, with some proteins more or less closed. It's kind of bonanza. And of course, one thing, in structural biology, many proteins share the same fold and almost the same structure. They have different very different functions. Many proteins, of course, have autoinhibitory mechanism this or that way. So there are some like golden principles that slowly we discover. What I want to show, I already mentioned what I see in sequence alignment, that almost all residues in said domain are exactly the same. So what we observed just titrating our protein with the ligand, we realized that there are some distant residues that essentially show significant chemical shifts. And they are not even first, not second, not third. They are like fourth, fifth, if we look like residue sphere behind the ligand. And these residues actually are not identical between all three of them. They differ between NSD2 and NSD1. And what we did, we just simply mutated them. So we took two NSD2 residues from NSD1 and from NSD2 residues. And what we found, we inverted selectivity of the ligand. So that's actually a beautiful experiment because it shows you like inverted engineering, that by changing some distant residues, you might tune the dynamics and behavior of the catalytic site and cofactor site that are identical among proteins, and then it will change preference for your ligand. Of course, a little bit of potency was lost, but still, the new NSD2 with NSD1 residues was not preferring this ligand. The better binder for this ligand was NSD1 with NSD2 residues. And then, of course, dynamic pattern changes of the site. So let's now talk about ASH1L. That protein is a little bit of peculiar because it gave me a lot of headache, and I think it will continue doing so because I still study it. So it's a kind of a big multi-domain protein like all of them. And the catalytic domain with this extension post-set 32 amino acids, orange one, is the same active like a full-length protein. Even in the cell, they reproduce the same in vitro, the same pattern of kinetics similar and products like in the cells. So it's okay to study the shorter version. They methylate lysine 36. That's kind of interesting because it's buried in the DNA nucleosome in contrast to lysine 27 that is easily available. So to study these proteins, you need full nucleosomes. Okay, let's move further. ASH1L has this N-terminal intrinsically disordered region with this FQLP motif. And when I started to study this protein, again, what I mentioned in one slide was it's good to know the target if you want to study, and it's good to also know the function. So that time I was thinking the target is only ASH1L. And when we started to work on it, NMR, ligand, structure, assignment, trying to crystallize, we invested a lot, mutants. Then this PhD student came to me and she said, hey, look, there are these papers and they say you need this MRG protein to activate your protein. I don't say I took it easy because that was essentially telling me that we have a problem. But then she was less panicking than me because she told me, look, this MRG is kind of weird. It binds FQLP motifs. Let's look at the crystal structure soon, but she somehow didn't want to believe that binding to intrinsically disordered part of protein, other protein, will release autoinhibitory loop and allow methylation. So, and also she did something I didn't do reading, that MRG15, it's not MRG protein. MRG protein is just fragment, one domain of MRG15 protein. MRG15 protein has this orange chromodomain. So what she did, she just simply downloaded all the structures that were published and just made this figure and showed me, hey, look, Łukasz, something is fishy. This MRG domain, first of all, it's not full protein, it's just the part of domain. It can have different conformations, but the most important is the H1L. Once it crystallizes with closed autoinhibitory loop with some cofactor, once it crystallizes with not resolved autoinhibitory loop without cofactor, and once with cofactor with open autoinhibitory loop. That's not normal. Still, I didn't know what she wanted to show me, but then she showed me this spectra. Look, we don't see 1/3 of the signals, even half of the signals for H1L. Even if we saturate it with cofactor, we still don't see 1/3 of the signals. And she asked me, do you know what it means? And then I didn't know what to answer, but I think there is dynamics. Yeah, and that fits why we can crystallize whatever. But what was more important, she just took two constructs and checked, okay, let's take the H1L and add only MRG like it was crystallized, no activity, function. So there is no sense in studying proteins if we don't validate at least the basic function. And then repeated experiment with full-length MRG protein with chromodomain restored activity. Okay, then I knew that she will get PhD because definitely she was much smarter than me, but I didn't know when. But then we started like regular studies. Okay, let's compare. What is the interaction of H1L with MRG and MRG with chromodomain? No difference. Okay, good, fits. What's the interaction of our MRG15 protein with nucleosomes? Like 60 micromolar. Not too bad. What's the interaction with only MRG protein of nucleosomes? Like 300 micromolar, so almost no interaction. What's the interaction of H1L with full-length protein with chromodomain? 6 micromolar, and that actually explains activity. And what's interaction? Yes? Ah. So, and what's interaction between H1L and MRG domain? The construct that was crystallized, no interaction, what explains no activity. And then we measured several other combinations, and everything started to add up to one nice story. So these are like experiments when by mutagenesis we validated that mutations in the chromodomain, this orange one, change activity on nucleosomes or affinity to nucleosomes, just to confirm that binding to nucleosomes to the substrate recruitment comes from chromodomain, not MRG domain. So if we don't know what to do, of course we do NMR. So we prepared H1L long construct with our extension and terminal domain that binds MRG. We did titration with MRG. We see beautiful tryptophanes. We see beautiful change of flexible parts. But I already mentioned to you we have a problem with TROSY spectrum. We have ongoing exchange in the set domain, so we don't see 1/3 of the signals. What to do? NMR is beautiful. If proton and nitrogen doesn't help you, ask proton and carbon because they are a little bit different time regime. And maybe they are not broadened beyond detection. And in this case, that was like a great idea because looking at metal groups, we still could validate that flexible metal groups from the intrinsically disordered part react with the MRG domain, but set domain, so folded groups, don't react at all, even if you do MRG or MRG domain. So that was a beautiful mechanistic confirmation. We also checked how binding, that was like a golden test to see if cofactor site an area where we design ligands is affected by MRG or MRG15, and we found that cofactor binding is essentially not changed at all. So that was like trying to kill the topic. So another thing what we did, we checked if ligands that are developed, different ligands from collaborator Tomasz Cierpicki from University of Michigan, if these ligands bind, with any difference. We didn't see any difference in potency of ligands, regardless if we have complex, we have full complex or not. And of course, doesn't matter if we have full length or part of protein, we reproduce exactly the same complex with ligands. So that was like one of the topics that I like to show because it shows really detailed investigation, combination of different techniques, and at the end, NMR for such dynamic systems was the most helpful and allowed us to distinguish if there are any allosteric structural changes. And it helped us to build a model that Ash1L protein is recruited by this MRG by FQLP, and all of this is recruited by chromodomain to nucleosome to do the job. So what is interesting, that's so dynamic system that we still didn't manage to get it on the grids for cryo-EM. We see all beautiful nucleosomes or beautiful blobs or just nothing. But you can see nucleosomes look awesome, at least. So we can prepare them. Here I show the slide that is kind of also emphasizing the big challenge we have with drug design. As I told you, Ash1L protein, all what is gray, what around, we don't see on the spectrum, and these are interesting parts of the protein. It's catalytic autoinhibitory loop, catalytic site, cofactor binding site. And then we measure dynamics, so methyl groups that we see with the empty apoprotein. We add ligand and essentially nothing changes. So we still don't see signals. It's still very dynamic with the ligand. On the contrary, when you crystallize the ligand, you get so beautiful density, so beautiful structure. And when you try to design anything based on that, you are not moving anywhere. You are not getting better ligands. So that's the question. Do you need to be really one structure to do the function? Do you need to really be rigid, or I don't know, that these are difficult questions. That protein, I told you, it's a headache. So, but that's a beautiful example. The same system, depending how you fossilize it, so crystallize or stabilize in solution in the buffer pH with TSEP antioxidant and excess of ligand, it tells you different story. So you need to understand it all. So that's why you need to integrate more techniques. So now let's talk about a little bit more about drug discovery. Why? Because why we do all of this structure biology? Not to collect all of those structures and be Carl Linné of describing new specimen of a protein, because it's kind of boring now, especially with the advent of prediction techniques that are getting better and better. We just want to do something useful. So we talked already about the left case when we have like drug binding site and we want to target it with some molecules. So usually computational techniques got so good today that when there is no major structural rearrangement after binding to the cleft or cavity or existing hole of the protein, they can propose quite good chemical space that potentially can be active, and usually it works better and better. So that's, we can say, easier, not so easy, but easier case of drug discovery, and it works pretty good. So the biggest challenge today, and usually $1 billion question, is protein-protein interaction surfaces. These ones are quite challenging, difficult, because usually they are big, they are flat, they are not really interesting from the point of view, like they don't have too many cavities or holes where we can stick some small ligand. And ligands cannot be also too big because they still need to have drug-like properties defined by, for example, like old good classics, Lipinski rule of 5. So that limits the size to around 500, 600 Dalton. So another group of proteins, like my pet proteins that I really like in the lab, and these ones are really amazing in the NMR magnet. They give beautiful spectra, but they share similar feature. There are different transcription factors. BTB, it doesn't really mean born to bind, but I think it's a very catchy nickname for the family of proteins. They are considered and they are largely undruggable transcription factors, and often they are linked to many diseases. What's exciting is there are 80 of them with zinc finger domains and over 350 of proteins in the human genome with BTB domains, and they are basically highly conserved, what we can see from the overlap of 3. What is exactly the same situation? The same fault, more or less, or very, very similar with primary sequence, end up with absolutely different functions. So we can still predict the fault, but will it explain details, essential details of mechanistics? That might not be always the case. So, of course, if we have proteins, we can ask question, hey AI, can I target any of these PTB domains? And the answer of AI is yes, you can. Where? Wherever. With what? With whatever. Great. So then we do experiment. So in one studies, the 3 proteins were prepared and fragment-based screening by NMR was done. So we use like small fragments. These are not yet drug or drug-like molecules, but small, usually weak binders. And then they are titrated over the proteins. We monitor with carbon, proton nitrogen, proton carbon spectra. And we find some hit rates. Unfortunately, not for all of them. Fortunately for one, and surprisingly too many than we expected. So from 1,600 ligands, if it's 7% hit rate, you can clearly see how many ligands we found for one protein and literally no ligands for others. The question is why? Yeah, but here we can ask this question, right? So why predictions were telling us everything can be targeted, and when we do the screen, we find nothing. So the answer is, okay, prediction is based on static structure, what obvious. These proteins still have some surface, and based on some analogy to one protein, for example, BCL6 that has some ligands in one place, of course, we can put them now in the same place because the fold is similar in every other protein, right? So what we started to do, again, if we don't know what to do, we do NMR, and if AI didn't help us, we use NI, so natural intelligence. So then we ask another PhD student, hey, that's a very interesting project. Of course, I had totally no idea what's behind, but let's study it. That can give great results. So after completing all of those relaxation data, we figure out that, of course, structure is highly similar. There are the same motifs, fault, but there is some peculiar and very interesting subtle behavior changes. So what we found from chemical shifts perturbation, we figure out mapped where the ligand is binding on the surface of protein, but we also found that there are some slow collective motions quite nicely overlapping, so exchange Rx, quite nicely overlapping with the protein, while Kaiser and LRF didn't have anything. Okay, this is correlation causation, so we pushed it further. We also started to look at the structure in solution, and we figure out that there is like low populated, like 5%. Second conformation that is open, that exposes hydrophobic residues. And that nicely correlates to what we found, that this protein is unique because only this protein among all tested has additional motions. And this we can come back to one of the previous slides, that one dominant persistent structure is not exactly what might underline some function or opportunity for targeting, because these two structures just interconvert within the conformational space. What is important, of course, if the project is hot and interesting, competition is also on it. So in the meantime, there was a publication from one group, Crystal Structure, of the same protein MEIS1, but co-crystallized with linear fragment of a very huge machine like Hui-1 E3 ligase that actually MEIS1 is recruiting to regulate MYC transcription factor, what has relevance in cancer. But what was important here, that was for me like, oh my, I was thinking this protein has this dynamics just for me because I found those ligands and I can develop maybe better drug candidates or degraders for this protein. But it turned out this dynamics was not for me. It was there just to recruit this E3 ligase to make MEIS1 different from other proteins. So evolution, then I started to understand what I heard many times, that evolution likely is not shaping only the fault, but also tuning subtly alternative conformation space that are context dependent to better fulfill and grab the function. Still, I don't have answer for the question. It's better to have two proteins separately evolved to do two functions, or one protein that for 1 or 5 or half % of time changes the conformation and then makes other function. I don't know the answers, but this is what's going on, and I think it's exciting. So, of course, what we did next? We did an experiment. We took the protein, it has dynamics. We added the peptide, we measured relaxation, no dynamics left. So this dynamics it was not for me, for ligands. I found and others can find. It was existing there for a while just to recruit this linear peptide with E3 ligase. Of course, we still have weak ligands, but what you do experiments. We can check if these ligands just extract, compete out the peptide from the protein. Of course, they do. They are good starting point for development. So this is more or less what we are doing. And like always, like in interesting cases and difficult examples, crystallography, we cannot crystallize. NMR, it's not too many constraints data. So then usually to have something beautiful, you need to combine two things. And as I mentioned, we need all techniques plus some modeling. Then we can get the pose of the ligand. We can propose analogs. Why we do it? Because MEIS1, as I mentioned, regulates MYC. MYC, there are some direct targetings of MYC now. These are protein peptide-like things, usually 4-stage of solid tumors, first clinical trials. Let's see how it goes. But small molecules are not really recommended, and MYC is so important regulator that whatever you do to MYC, essentially you just kill not only the cancer, but also the patient. But what we found by targeting MEIS1 or by removing by CRISP MEIS1 from the small cell lung cancer patient cells. Actually, what you see on the left in collaboration with Paweł, he's now in MD Anderson. Actually, we studied in University of Warsaw together, so the time is also crazy. That's why I mentioned that even being in the US, across the world, I still work with people I met and collaborated in Poland, and it still continues. So what is important? Small cell lung cancer is essentially the worst one. There are no treatments. These percentages are really scary and no one really makes it. And it's commonly amplified MYC connected with it. So if we target regulator, maybe we have kind of good genetic validation here of a target for this particular cancer. That's why we are pushing one part of the project lab to develop more potent ligands inhibitors of this Hui recruitment. And of course, like the big picture is now with NMR and dynamics, alternative states, we can expand the druggable genome if only we can know which proteins have alternative conformation that exist, that are functional, that are ligandable, that we can target. But yeah, but at least it's a beautiful vision. So what I was telling to you is essentially nothing new because dynamics is important in life, what we know. And actually, this is one of my favorite paintings. So you can imagine it's 1434, beginnings of Renaissance. And then we know what was before, those flat Middle Ages. It's like those flat fossils, flat specimens. It was flat. Here, for the first time, we have one of the first attempts of the painter. We see him in the mirror that is ideally curved. There is no single mistake with light entering the room, with geometry, with perspective. It's excellent. So the story: the painter with his wife visits his friends, Arnold Fini. Those were the beautiful times to marry. You didn't have to have priest in the room, and it was starting already. So we see that there is a message. Happy couple, they are about to start a new chapter in the life. But then when the painting was finished, about to be finished, and this life is not linear, life is not frozen, life happens. So there is a message in the mirror that things not always move so smooth or not stay as pictured with one snapshot. So I think it's genial. So thank you so much for your attention and discussion. So I hope I convinced you that proteins is all we need and we need to study them more. This is what my cats think, and they are now sitting here around about what I'm studying. This is like, yeah, coffee. I still have some coffee. I hope you too. And just a summary slide at the end. Thank you.
Thank you very much, Łukasz, for a fantastic talk. And we are open for discussion. If you want to ask questions, please turn on your microphone and, if possible, camera. I especially encourage students to ask questions. I see some students that work on protein NMR, so I think they have very good questions. Let's give them a moment.
You can win a cat.
Cat not, but a coffee mug, for example.
Yeah, that one was good, actually.
Yeah. Do you have any questions? Okay. Looks like probably maybe I'll then ask some warm-up question. So when you study the ligands, do you have some closed library of molecules that you use to study the interaction of this protein, or it's more Wild West, let's say?
Yeah, so that's actually a very good question, and that's part of the workflow. So depending on the system, sometimes we just know that there are some ligands that they are already published. They are in clinical trials. Like we now work on P53. There is this Y220C mutation, famous one in 1% of all cancers regulated by P53, but P53 is 70% of cancers, so still worth studying. So there are like second phase clinical trial ligands, and now we study them because we have idea how to make them better. So we start from existing compounds. If you have your target, you know that, okay, there are nothing targeting. Of course, it's good to start screening with high throughput screening, drug-like libraries. You might find something, many false positives. Life is hard. You need to have a working assay. It gets expensive, big numbers. You need to have working microassay. Sometimes when you make it small, it doesn't work anymore so well. Another point is if you want to study by NMR, fragment-based screening, it can be protein observed, ligand observed, or you can do crystallography. If you have nice crystals, you can soak your protein with soup of ligands. So this is how we work. And yes, usually there are libraries. So depending what you study, definitely you don't want to, for example, have kinase inhibitors in your, like drug-like kinase inhibitors in your cocktail because you don't want to have off targets later. You might find a beautiful ligand, but you don't want it to kill randomly kinases in the body, right? So there are some kind of guidelines. There is some common sense. But yeah, you are right. You found it well. It's usually bonanza, and it's more discovery than design.
Okay, thanks. So now maybe someone is brave enough to ask questions. Let's give a moment.
Can I ask to the board, Tomasz? I don't believe, Tomasz, you don't have questions.
Yes, yeah. Well, maybe. Okay, I think so. On some slide, you showed a molecule with fluorine groups. Could you
Oh,
comment on this? I mean, is it possible to utilize this in NMR or something like that? It
Tomasz,
was CF. It was CF3.
yeah, exactly. So I have to tell you that fluorine is something that we truly, truly like in our libraries and compounds because you don't need to worry with water suppression. You don't need to worry about overlap and on the proton spectra. And that's a very cool probe. You can do like STD fluorine, water loxy fluorine. If you have, sometimes you have more than one fluorine. You have CF3 group, your favorite, and you can have some aryl fluorine. So this works usually good, but there is one limitation. You need to have probe that can measure fluorine. So, and with current Bruker spectrometers, it's not enough that you tune the proton channel like left and you get fluorine. It's no longer there. But still, I think we have here 500 broadband probe. We have quite sensitive fluorine. So this is what we are doing. We have ligands. We do interaction. You can get really a lot of information.
Okay, thank you.
Okay. Do we have another
Hey,
questions?
hello.
Oh,
Hey.
good.
Yes, I
Cześć,
have a
Krzysztof.
question.
Yes.
Hey, Krzysztof. Yes.
Hey,
Hey, Mateusz. Hey, Łukasz. Yeah, so yeah, I only had audio for this talk because I had to start my travel back home. But I
oh,
wonder, yeah, so maybe I missed something that you showed on the slides, but I wonder what changes do you expect for these specific systems in terms of ligand binding or in vivo? I mean, when you switch to actual in vivo cases, or when you would switch instead of in vitro studies. So do you expect any dramatic changes for some of the systems?
So basically, that's essentially the first thing. Thank you for this excellent question. That's the first thing that needs to be checked and validated from the beginning. So we need to really understand the system. So in this case, I'm quite of boring because I work only on methylation. Why? Because this is something you can really validate with Western from directly from the cells from your models, animal models. So you can check. You can also check connected genes activations. For example, with NSD3, when it's hyperactive, you regulate mTOR pathway and MYC. So you can check. These are not direct things, but directly you can check the product, like demethylation on chromatin. So that can be checked. And you add your ligand, you can see if it goes down like you want or up. So there are direct and indirect things. Now the question, if we have ligand and it works beautifully in vitro, you are absolutely right to question if it's going to work in vivo, because in vitro is isotropic solution. We don't have membranes that we need to cross. We don't have plenty of other things that will stick, and we don't have plenty of off targets, what I mentioned already with gently with kinase inhibitors, that if you screen for compounds, first thing you do, you kick out everything that looks like kinase related compound, because even if it works with your peculiar target, you don't want it to have any off targets. So probably your ligand can be effectively diluted even if it doesn't do anything wrong in the cells or in the organism. So yeah, that's a good question, and I have to tell you that's why developing compounds is really tough, and 95% of them will crash.
Okay, but in the whole ligand binding workflow, when you study new ligands, where are in vivo studies? Are they in general prior to NMR or after NMR? How is it?
So,
I mean, when you consider multi-technique approach, so to say, and
Mhm.
multi-sample.
Yeah, so essentially what we do, NMR and structure biology, biophysics, solution biophysics is before, because we want to find compounds, we want to get them to some potency in vitro that we know, okay, it's binding, it's selective among the test groups. Like we have, for example, NSD1 to 3, we want it to be selective to 2, we got it. And then, of course, we start with cell lines. and we check exactly the same product. So if we check them, if they bind, usually binding doesn't help. They need to do something with the function. So we look at, these are enzymes, so they have enzymatic output. So this helps. So we check enzymatic output in vitro. And of course, in vivo, as we already discussed, in the cells, like methylation mark, lysine 36, histone 3, demethylation antibodies, boom, they go down, we are happy.
Okay, thank you.
Thank you so much.
Thanks. Bye.
I hope Krzysztof stopped cycling for asking questions.
Yes, I did. Yeah, but I couldn't watch slides. Sorry, at least half of them. Yeah, but the talk
come
was
on,
very
I'm so
interesting.
I'm
I got.
disappointed.
Yeah. I will watch on the YouTube later, since it
Thank
was
you.
recorded. Yeah. But audio was great. So thank you.
Thank you so much.
Okay, I think it's our first seminar on bike. Okay, but so students, last chance for asking question and winning, or not winning, but participating in the game.
So how about if no one asks questions, you can
Well,
send back to me.
actually, I have also one question.
You are not a student, but okay.
Yeah, I can be, Okay, so do you think that would it be possible to hyperpolarize all these small molecules by photokidnap?
You mean what, like parahydrogen, or how to?
No. By photokidnap, by light.
I didn't think about that.
Because I think so. They are. I mean, they are. I mean, very similar to. I mean, I don't know. Maybe. Yeah. This is my question actually.
Okay. I have no idea. I would be very happy to read a bit more. So maybe you can drop me a line with some papers. That would be super cool.
Well, actually, we are not doing this. I think so. Ronald Riek is doing something, with photokidnap and also proteins or something like that.
That's
Yeah.
very interesting. Yeah. So all those NMR guys in Switzerland. Yeah. So they have quite interesting things. So. Yeah, but down to earth, you are always limited, but by what you have in your facility. So
I mean, the photokidnap facility is actually, depends. It is actually, I would say, relatively simple. All you need is just, it is not dynamic nuclear polarization or, something like that. It is, I would say, $10,000 probably is maximum, and you can have everything.
Oh, okay. So that's something I would be very happy to have a closer look into.
Yeah.
Yeah, thanks
Okay.
for mentioning.
Okay, so the very, very last chance for students to ask questions. If not, okay, they don't want so. So thanks everyone for joining us. Thank you very much, Łukasz, for giving this fantastic talk, and everyone for the discussion.
Always happy. Thank you so much. I hope everything is fine, and
okay.
Ciao.
And I'll just show what's coming on soon. So in 2 weeks, we have 2 talks. One is the student talk from Sylwia Jopa from University of Warsaw, and the second is from Jan Stanek, also from University of Warsaw, however, different faculty. And we're going back to 12 p.m. So thanks everyone for joining us, and see you in 2 weeks.
Thank you very much. Bye-bye.